again, it's my pleasure to introduce to you again Jingyuan, who's going to be talking to us in this next lecture of hers on laminar functional connectivity.
Okay, great, thanks.
I can give you this. This pad is a little bit funny. Okay, there you go. Use the left arrow. Yep.
Okay, I start by just saying we all know fMRI is a very powerful tool for mapping brain functional architecture. I think it's compared to other imaging modalities, it has a particular advantage of covering a lot of parts of the brain, so you can really look into the interactions of different brain regions. But then we always have questions. Now we know how the different parts are kind of wired to formulate the brain functional architecture. So what are the causal influence of different regions kind of exert to the others? And on the other hand, how do we know when you have two regions kind of to have a peer or apparent connectivity, whether they arise from direct connections between these two regions, or they just receive intermediate connections from other brain regions? So we actually have already learned a bit about this effective connectivity from David Norris' talk on day one. It's just quite straightforward idea where naturally now we learn that laminar fMRI can provide some canonical circuitries to kind of inform those directionality information so that we can leverage them to infer directed connectivity in fMRI. So that's kind of the general idea for this talk. So first, just to bring everyone on the same page about the definition of functional connectivity, and then we'll go into details with the theory and a few examples of how people do exciting exploratory analysis of effective connectivity with laminar fMRI. And finally, I will just highlight a few small challenges, or not small challenges, some challenges, particularly for laminar connectivity analysis. Okay, I first want to especially acknowledge Renzo Huber, who actually shared with me lots of those slides for this laminar connectivity analysis. Okay, before going to functional connectivity, I think if you talk to people with traditional neuroscience backgrounds, when they think about connections, they always think about regions with really physical connections. So probably you'll hear the terminology called neuronal cartography. So basically use direct viral tracing to identify those neuronal connections. So we actually probably see some of these images from Yosin's talk in day one. And also you may hear people are talking about structural connectivities, really using diffusion MRI to make sure different regions have those white matter fibers really kind of tracking these different brain regions. So these kind of the two ways are really ground on these physical connections of different regions called it a connection. And for fMRI, it would be quite surprised. It's quite different definition. So for instance, this just raw fMRI data evolve over time. Actually, people's interest in this kind of intrinsic spontaneous activity dates from their earlier observations of EEG and the PET. So basically, they account for the majority of the brain's energy, about 20% of it. So it's really have quite a rich information in this ongoing dynamics. But now we have fMRI will be able to track these kind of coordinated behavior of these networks. What's kind of interesting is people realize there are really ordered spatial organizations of these spontaneous activities. So for instance, if you look at time course from some anterior portion of the brain and some time course from the posterior portion of the brain, they're just not random noise. They really have those synchronized behavior. So the definition of fMRI is quite surprising. It's purely based on the temporal synchrony of the activities across different brain regions. You may see earlier actually her work from Peter Pentini, where they use a correlation analysis really quantifying this kind of thing. And then, so the first kind of really showing this fMRI of those resting state connectivity where it's just when they compare these patterns, putting a seed in the sensory region, the networks kind of with the strongest activation really resemble those activation patterns where you do a finger tapping task. So, and actually the organized behavior not just limited to the motor network. If you parcelate in the brain into a lot of brain regions and just look at those inter-region connectivity, you can see that it really organized into different kind of parts where some regions have very strong coherent activities compared to the others. So that's why over years people have identified more than 10 kind of resting state networks. They operate simultaneous in the resting state brain. So this is just one of these kind of early postulations where you can see the different networks. But nowadays, with high spatial resolution and dense sampling of individuals, we are going to get more refined architectures of these networks. And how do we derive these networks? Some like conventional way is quite straightforward. One is called like independent component analysis. So it's basically doing 4D spatial temporal decomposition of these four-dimensional fMRI data and assume some additional assumptions on the spatial independence and sparsity of the data and decompose it into different networks. And others will just be direct clustering or cross-relation based on similarity of these time course. Those voxels have very synchronized or similar activities. They just belong to one network. So again, this is just one earlier canonical parcellation. But when we see these quite interesting organized activities and networks of the resting state networks, people always wonder whether they arise from neuronal origins. So I think these kind of controversies even persist till nowadays. But over time, there are actually many studies using complementary measures already demonstrate a kind of neuronal relevance of these resting state networks. So for instance, this is an MEG study where they can see some like organized MEG activities just look a little bit like those resting state networks people see with fMRI. And here is another example where I use intracranial ECOG data, and you can see the envelope of very high frequency power really shows synchronized behavior in the anterior and posterior parts of the brain. Here just another kind of interesting example where in the mice data, I think it's super cool. You can have direct recordings of the calcium data, and when you contrast it to the simultaneous hemodynamic data, you may notice a little bit delay, but really kind of there's lots of ongoing coherent activities between this calcium activity and hemodynamic activity. So providing strong support to this neuronal origin of this kind of spontaneous activity. But while it kind of probably have a neuronal origin, but doesn't mean we have very clear understanding of the neuronal mechanisms giving rise to this spontaneous activity. I think this is just one study I really like kind of highlighting the complexity of efforts trying to link this functional connectivity to these kind of physical connections of different parts of the brain. So in this study where the authors kind of section the corpus callosum, and ideally you are going to break the interhemispheric correlations. So what is shown here in the top is like pre section, the kind of whole brain-wise connectivity matrix, and the bottom shows the post-section. And ideally, you will see after the section of the corpus callosum, you're going to see the connection separated into the left hemisphere and the right hemisphere. And what's the difference among all the three monkeys, which is kind of each column is one monkey, is that for the monkey N, they kind of sectioned most parts of the anterior commissure, but with a few kind of fiber spares. And for the middle monkey, it's really kind of entirely sectioned this anterior commissure. And then the third column is a monkey where they leave the anterior commissure intact. So what's interesting is that while you kind of cut off all these corpus callosum, but if you leave the anterior commissure kind of intact, lots of the connectivity is still preserved. I think this kind of just one example highlighting it's not quite straightforward to directly link functional connectivity to structural connectivity. And then temporal coherence among regions is really just a quite simple but hard to interpret phenomenon. Yeah. Okay. But while we don't quite have a very clear understanding of what functional connectivity is and the underlying mechanisms, but it doesn't hinder people's interest in these particular domains. And you can see the publication really kind of grow exponentially over years. I think probably due to multiple reasons. On one hand, it's quite accessible. It's so easy to do this simple, robust correlation analysis. But on the other hand, all those phenomena of correlated networks and how it changes under different conditions, just such a robust phenomena, and people can easily replicate and always see in different conditions. So over years, people have found like this connectivity can kind of correlate with aging and different mental illness, like intelligence and all sorts of behavior. And also the connectivity analysis just not just limited to resting state conditions. And then these connectivity structures transcend across like sedation and sleep and also different cognitive states. And very recently, there has been growing interest in pursuing naturalistic stimuli. And compared to spontaneous resting state activity, it apparently has several advantages. For instance, it's in a more controlled manner. So you really kind of have an imposed stimulus. And also you can do multiple movie watching or whatever clips to average and to boost your SMR. And it also opens up the possibility for you to perform inter-subject comparisons to look at our communications. Okay, so we have learned about this kind of functional connectivity between regions, but in parallelly, they're always interested to see what are the directed information flow kind of transferred between regions, because the direct correlation doesn't provide such information. And then there has been some approaches people tried. First is kind of along the causality analysis. So basically, based on the timing differences across different brain regions. Put it simply, if a region's activity precedes the activity of another region, so likely probably has a causal influence on the later regions. But for causality analysis, apparently has some limitations because for fMRI is really built upon these hemodynamic changes triggered by neuronal activity, and there are huge inter-subject and inter-region variations of this hemodynamic response, and how to address this kind of inter-regional variability of these hemodynamic changes are always a concern for this causality analysis. And another approach people tried is using direct neuromodulation. So for instance, using TMS or deep brain stimulation to excite or inhibit a particular brain regions, and then check the activities in other brain regions, and using this way to probe the causality in a specific brain circuitry. It also has some limitations, especially for human studies. It's hard to make it quite invasive. And for these non-invasive stimulation approaches, really suffer from unclear mechanisms and also relatively poor spatial specificity. Oh, okay, thanks. Yeah. Okay. Okay, you hear me okay now? Okay, great. Yeah. So here, just like for the second part, we kind of discuss how laminar fMRI can provide an opportunity for us to map these effective connections across the brain. So we have seen, I think, this kind of canonical cortical microcircuit from several lectures throughout this course. And then this actually given us an idea of inferring these kind of input and output and feed-forward and feedback connections in different brain regions. So for instance, for two regions, if you want to identify the direction of the connections, you can plot their layer by layer correlations. And if you see like the correlation strongest, for instance, the output layer A to the input layer B, and then you can actually identify the direction flow and vice versa. And also, by checking the correlation, whether it's strongest in the kind of layer for the input layer, and then probably kind of can suggest it's kind of more like a feed-forward connections. If it's kind of superficial or deeper layers, probably also can suggest a higher probability of receiving a feedback connections from the seed region. And another advantage is like for two regions, if you want to check whether their interactions are mediated by a third region, we can also check the layer profiles or layer connectivities with the third brain regions, and if the connection are strongest in both the layer for both the regions, probably can suggesting their correlations arise from a common source. So essentially, these kind of layer profiles really provide a potential opportunity for us to disentangle all these kind of feed-forward feedback connections among regions, and also potentially elucidate these direct information flow that can mediate this kind of macroscopic functional connectivity. But on the other hand, effective connectivity doesn't or there is no strong support for why they would be aligned with functional connectivity. And many ideas people use to compute the functional connectivity probably can inspire to do similar ways to extract those effective connectomes. So for instance, we always use which regions are strongest correlated to a C region to infer those functional networks. And similarly, we can use this kind of effective connection approach to identify those regions receiving the feed-forward or feedback inputs from a specific seed. And also, what are the cortical parcellation will look like if we, instead of based on the functional similarity of activities from different regions, are based on the layer connectivity profiles. And just like we do whole brain functional connectivity, whether we can infer these effective connectomes from the whole brain layer profiles. And hopefully, I think as all these kind of findings will be replicated and then can test their own ways, they will kind of pose new clinical biomarkers for our like many studies. And also, as if we have better understanding of these signal mechanisms, ideally at some point it will help gain insights into the neuroscientific and clinical questions. But however, all these kind of great opportunities were built upon we really have a perfect laminar fMRI data set, and also the layer models are really kind of is universal to all those cortical regions. But now let's just assuming that would be the case, what the kind of interesting analysis we can do with this laminar connectivity analysis. So this is just like first one of those studies where showing this initial validation from Renzo, where they trying to infer these effective connections in the visual system. So we know that in the visual system, in primary visual cortex, V1 is receiving the feed-forward input from the LGN in the thalamus. And also for higher visual area, it's kind of sending feedback inputs into the V1 area. And then we can use laminar fMRI to really test whether we can see these kind of effective connections. So in this study, so basically they marking all the seeds in the LGN, which is the green color, and the blue in primary visual cortex, and also the yellow ones in the higher visual area. And then we just check the correlation of the layer profiles in V1 respect to the seed in LGN and the seed in the higher visual area, and to see whether the patterns align with the hypothesis. So here just shows the connectivity layer profiles of the different layers in V1 respect to the seed in LGN. So really can see a kind of peak close to the layer 4 area. So it's kind of supporting a feed-forward connections from LGN to V1. And if you check the connections of V1 to higher visual area V5, and then you can really see this double peak pattern with the superior and a little bit deeper layers have the strongest activity. So this is just a very kind of encouraging case, like supporting like layer connectivity really can support those canonical circuit people are seeing in these animal models. And here is just another example extending to the motor cortex. So here we're using a seed in the S1 area, and then from these animal models, we know it has the inputs to the superior layers, superficial layers of M1, and you can clearly see that in the layer profiles really have the peak, upper peak in the superficial layers. And if you take a seed in the premotor area, and then you will see a quite distinct pattern where you're receiving feedback inputs to the M1 area. And then just here, just shows a comparison with you choose a random seeds in the different brain regions where you don't see a very clear structures of these kind of layer profiles. And after kind of testing these kind of primary sensory regions, you certainly have interest to extend into higher cognitive areas. So this is kind of an interesting study where recently kind of started to looking at the effective connectivities in the deep neural network. It's one of those kind of strong active networks in the resting state. So they're taking the seeds in the prefrontal cortex and also the posterior parietal cortex. So if you use a kind of a seed, the seed in the kind of parietal in PCC region and to the MPFC, and you can see the profile in the frontal area really peak in the kind of middle layer, suggesting there is a feed-forward input to this kind of frontal area. And reversely, if you put a seed in the anterior area and then look at the layer profiles in the posterior area, it has a peak in the superficial area, kind of probably indicating a feedback input to this kind of posterior portion of the brain. And certainly, we can always extending this analysis to the whole brain scale. And then this just a nice images kind of where if you zoom in the details, you can already see in the different brain networks. For instance, like in the visual area, you can already clearly see these kind of double stripe patterns here. While in the PCC area, you only see a kind of nice single stripe. And here, just the different networks can already directly see from the raw activation patterns of different networks. But if you go into specific networks, we can clearly see these kind of layer-dependent interactions of different regions in the system. So for instance, here, just the visual area, and you can clearly see like for V5 and V1, the strongest correlations are in the superficial and also deeper depths. And you can extend it to different parts of the brain of the networks. This will just give you a way to achieve this kind of whole brain scale connectome patterns. So I think by far it's really kind of just the canonical way you have different patches of the brain and you start going to see the pairwise interactions of these effective connections. But I really have many interesting ways we can also check, as mentioned, whether we taking a seed, we can really to check which are the brain areas receiving the feedback input from a particular seed, and which are those regions receiving feed-forward inputs from a seed. So here just an example where going back to the visual data, if you have the canonical kind of assumption of the templates for feed-forward connections should be kind of peaking in the layer 4, and for feedback inputs were from more like superficial and deeper layers, and then we can basically taking a seed and then exploring this kind of hierarchy of the connections. So for instance, if you place a seed in the LGN, you can really see that in all those higher visual areas and the primary V1, they all receiving these kind of feed-forward connections from the seed. And if you move the seed to the primary V1 area, you can see those extra striate higher visual areas are receiving kind of feed-forward inputs from V1. And if you move the kind of seed to the further into the extra striated cortex, you can also going to see like if further along the cortex, they're receiving the feed-forward connections, but like in a lower V1 area, it's kind of receiving feedback input from the seed. So the pattern, if you go further into the striated cortex, you will see only very higher order visual areas receiving bottom-up inputs from the seed. Yeah, and I think that's kind of just shows the way you are checking how the profiles of the different brain regions relative to a specific seed. But for different regions, they're kind of receiving inputs from the entire rest parts of the brain. That also allows us probably to do other exploratory analysis, basically identifying whether kind of this region are dominated by receiving these feed-forward or feedback connections. So this is just an interesting analysis where the harmonics is basically defined which layer best represents the overall fluctuations in the column. So the idea here is just that across the column, they will correlate the signals to the mean of the signal in that kind of across column. So then you will get the profile of the correlation across different depths. The ones with a higher correlations basically suggesting they probably have the more dominating kind of behaviors of these kind of particular column areas. And then if you classify the brain according to the specific harmonics profiles, you can clearly see it separate into two parts, where in the anterior parts, it shows a pattern where it have like more increased or higher activities in the more superficial and more deeper layers, whereas the more parietal areas have the particular harmonics patterns more peaking in the middle layers. And also, while previously kind of we're talking all about this kind of layer-specific information about feed-forward and feedback connections, and each layer probably have its own particular connectomes. So here are just some early studies were kind of exploring, or you already heard from David Nar's talk, where they using generalized PPI analysis to explore those spatial clusters interacting with the specific depths of the region. And you can really see like for the clusters connected to the middle and deep depths of these occipital-temporal sulcus are quite like distinct, which I won't go into too much details. But there's a kind of interesting recent study too, where they basically testing a hypothesis in animal studies where they're showing the anterior and the posterior portion of the hippocampus. They're connecting to different layers of the higher cognitive areas. So the specific model is like for the posterior portion of the hippocampus is kind of the anterior portion of the hippocampus is correlated to the layer 5 of the dorsal attention network during the encoding process, while during retrieval process, it's kind of the posterior portion of the hippocampus is correlated with layer 2 of the deformal network. So, and what they're testing is whether they're parcellating the hippocampus based on the particular connections with the higher cognitive areas. They can really separate these kind of profiles of the anterior and the posterior portion of the hippocampus. And what's not shown here, they also show that the correlation of layer 5 with the anterior portion of hippocampus is strongest during the encoding task, whereas the correlation of the deformal network and the posterior portion of the hippocampus is strongest during the retrieval process, kind of impartially kind of supporting what people see in these kind of animal studies. And other examples would be also we can leverage the mesoscopic layer information to really kind of refine these network structures we can gain from the data. So for instance, these are just common brain networks for very standard studies where you extract the different brain networks of like the visual network and all those motor network and executive control network. But if you do further ICA into these networks, you start to parcellate in the different regions kind of related to different functions of the model of like hand and face in different areas. And then if you pull out the hand area, you can do further iterative ICA, kind of further divide it into more specific networks. And if you compare it with the activation map of like doing like index finger and the pinky fingers, and you can see it really aligned with them. And then if you do further iterative ICA kind of in the approach, you're going to have a finer divisions of the network. In this particular case, the separation was not kind of really match all these layer profiles, instead of based on the columnar structures kind of related to more specific finger tapping behaviors. So this is just an example where when you go to very high spatial resolution and have information gathered from different layers, you will be able to give like more refined structures of the data. And final example I'm going to show is kind of a study. I found it quite interesting. It's kind of using laminar fMRI, basically whether the additional information we gather from layer can help inform this intersubject variability we see in the data. So basically, this was using the naturalistic moving watching paradigm, which we mentioned earlier, because it has an advantage of really kind of performing the task in more controlled manner compared to resting state. And also for laminar fMRI, we need to average the data from multiple movie click, have the advantage of kind of, we can run the movie several times and then average the signal to boost the SNR. And then, so in this study where like, this is a paradigm similar to what people use in the human connectome data. And then the subjects kind of basis on the activity of the task positive network. They are separated into different networks, different subject population, and it would be interesting to see whether there are specific layer information given rise to these kind of distinctions of these time course people see in the different groups. So I think what Rezo did was they also collected the data from the using VASO in the whole brain layer fMRI, and then you get the kind of network patterns. So the analysis was kind of focused on the task positive network. So the basic idea is that when people are watching these kind of movies, there are some periods where like they will have very stable inputs of those visual in the low visual area, but there are also some scenes kind of involving very subject-specific cognitive behaviors. So the analysis what they're doing is like in the HCP data, when you break the subjects into 3 different groups, and you can see they have quite. Actually, the separation was based on the similarity of the time course. We can really see the group of subjects when they go to basic visual inputs, they have more stable or more consistent dynamics of these fMRI time courses. But when you go to other frames, for instance, in the kissing scenes, you will really see inconsistent behaviors of these fMRI activities across different groups. And then if you use this time course derived from the resting state HCP datasets and regress it against the layer fMRI datasets, you really can see a very interesting distinction of the layer profiles across the subjects. And then it seems like one group have very kind of much higher superficial kind of inputs here. And some interpretation seems quite interesting. It's also like when people watch a movie and then for the visual inputs of this feed-forward connection seems more consistent across subjects. But when you go to a higher areas and then you start going to have the feedback input, probably will be quite distinct across subjects. Okay. Yeah, and then I think another interesting thing I feel about this study is also they said when they try not correcting for physiological noise and not going to see much difference in the wave profile, that actually highlights a quite compelling analysis paradigms for this data because the time course they're deriving are not from the data itself or from a very distinct population. So yeah, I think it's quite an interesting concept to me. It's like if we do this kind of interceptor comparison or analysis, not simply just give you a way to infer this across subject effective connectivity, but also make it less vulnerable to correlative noise across layers and regions across the same subjects. For instance, like the physio noise we talked about yesterday. So you will circumvent these kind of caveats just by correlating signal from one subject to the other. And finally, we'll just quickly go through these kind of potential challenges specific to laminar fMRI connectivity analysis. So of course, all challenges faced by laminar fMRI studies will have similar concerns in the connectivity analysis as well. But then, especially in the case when we try to estimate the connections among different regions, so the limited field of view really will constrain the causality model where you have. And also, when you infer information across different brain regions, this sampling variability of these layers due to cortical thickness, inhomogeneity, and also the variant thickness of layers across subjects, this all going to pose concerns. And another one is, just like all the other studies, we are also neglecting the potential within column like neuronal communications, and also this activity are going to modulate it by or further blurred by interregion variabilities of those vascular layers across regions. So for instance, that meaning even you have a neuronal input here, what you actually measured data will have slightly distinct profiles. I just want to highlight this probably be more severe issue for resting state studies or in cases where you have minor neuronal inputs, because in those cases, the contrast caused by this baseline bias probably would be comparable to those neuronal or stimulus-driven effects. And finally, it's just that we really lack a universal cross-layer feed-forward and feedback models as kind of shown here. Like, for instance, there are some regions just not lacking this kind of layer 4. So in this case, the canonical model wouldn't apply. And also here, just an example where they're showing if you measure the post-synaptic potentials in layer 5 and layer 6, post-stimulus, they actually get faster than the post-synaptic potential in the layer 4, basically suggesting that deep layers can directly be activated by thalamus without the sensory kind of in the sensory processing. And another challenge is, I think this study kind of have an illustration of that is when we have this layer information, we're really going to have a lot of information on how we're going to do data mining and visualize these connectomic results. So for instance, here just basically showing within the deep neural network, the interactions among different brain regions, and the different wires here is kind of including the different layer information. It's already quite a complicated feature, but imagine we have a matrix and then expand it to the whole brain scale. How are you going to kind of measure this data? And finally, so I just want to show this image I really like, because on one hand, it really shows the layer profiles, which is kind of a right curve, like thick curve showing here, like across different cortical regions. So when you see it, it's just like you cannot pretend you never see this. So that actually kind of provides a case. It's like nowadays, for the most advanced neuroimaging techniques, we are able probably to see the layer-specific profiles of information that can only previously see in these animal models. So, but on the other hand, yeah, while it's kind of encouraging, it also does suggest how to interpret these patterns. Probably need a more detailed mapping of this cytoarchitecture and myelin architecture. But so even though they are validated in the sensory paradigms, probably when we extend it to other brain regions, we'll warrant a little bit caution. But this is just a still quite new field. There are many years to go to keep kind of refine and optimize and validate these things. But I think the bottom line is like, as long as we can see robust results and all those things can be replicated in different disease or neuroscientific conditions, it doesn't prevent us by leveraging this layer information to provide very robust biomarkers. And probably it's also the time kind of embrace discovery science. It's like, while we see this quite interesting phenomenon in human, we can always devise kind of interesting hypotheses and then validate those in those animal studies. Okay, so for a little bit over time, and then I would just highly strong to read the layer connectivity paper by Renzo and his colleagues. So this really kind of motivates a lot of these slides kind of shown here, and also they have an open data set. I think people can explore with that. Okay, thanks so much. Yeah.